So I will tell you a little bit about what I did in my PhD, about how genetics can actually help us in understanding invasive species. So maybe a little bit about me first. I like butterflies, but I haven't managed to work with them yet. And I actually started out with chemical biology in my bachelor's, so not really a straightforward way, because then I moved on to anthropology, where I worked with ancient humans. And then I also decided that was not really for me, so I decided to do a PhD in plant genetics in Norway. So that's also where I still am for a postdoc, so maybe I finally found what I want to be working with. So I use this lovely plant, maybe not that lovely, common ragweed, to actually do my PhD. It's a weed, as the name says. You can't smoke it, so. And it can, as you see here, it can actually get quite tall. So also on this picture, it's ragweed in the background, so it's actually taller than me. It can be up to 2 meters, or it can be tiny, like sometimes it's just like this big. And it's annual, meaning it only lives for 1 year, and then it dies as soon as the first frost comes. And also, is anyone here allergic to pollen? Yeah, quite many. You will hate this plant because it's the main cause of hay fever. And also it flowers quite late in the season, so it actually extends the pollen season by quite a lot. So a little bit nasty. A little bit about like what actually is an invasive species. So imagine you have the native range here. So this is where it's normally found. And this is also, coincidentally, the native range of common ragweed. And then you suddenly find it here in Europe. But you know, there's a giant sea between them, so it actually can't reach it. It needs help from humans. And because it can't reach it on a natural way, we call it introduced, because humans introduced it. This can be on purpose. Sometimes like plants look pretty, not ragweed, but you may want to have them in your backyard, so you introduce them. Or like sometimes we introduce species because we want them like to use them for pest control. That's like, you maybe know this famous example of the toad in Australia. So yeah, humans also did that. So this is then called the introduced range. And ragweed actually get introduced all over the place. So all this like orange things are introduced ranges for ragweed. So it's today found on all continents except Antarctica. And how did that happen? Well, the red here, it's actually the ship routes. So you see how interconnected the world is. So when you go over from one place to another with global trade and you have all those goods, then you may accidentally just bring a plant with you that you didn't want. So this is also what happened with ragweed. We introduced, for example, horse fodder from North America to Europe, and there was some ragweed in there. So then it just managed to grow in Europe. So well, it got introduced, but that's not enough to actually be invasive. So as I said, it only lives for 1 year. So it actually needs to establish a population. It needs to survive and grow there for several generations. And then it also needs to spread in the invasive range to be called, in the introduced range to be called invasive. And it also needs to harm the environment. So it does harm humans, of course, so that's already a good point for being invasive. But also, as you may see here, this is supposed to be a sunflower field. There are not many sunflowers there, because ragweed just took over and reduced like the yield of sunflower by quite a lot. And it doesn't only do that in sunflower fields. It also likes to grow in like soybean fields and other crops. So not so good. So it's an evil ragweed. What I actually did then, coming more to the science stuff, I took samples from all over the place in North America, where you can find it, and also in Europe. To actually then, we wanted to look at the genetics and how it changed, like from where in North America they actually came from and stuff like that. But then we have one big issue that like the modern variation does not necessarily reflect the historic ones. So this plant got introduced to Europe in the late 19th century, so that's a lot of time to already change. So this is also an example for like how the modern variation may not reflect history. Is anyone here lactose intolerant? Not so many actually, and that's because we're in Scandinavia. As you see here on the map, like the dark is like the percentage of people who can digest lactose. So it's really common in Scandinavia and the UK, and not really that common outside of it, and really uncommon outside Europe. So you may think it actually arose in Scandinavia or the UK, but that's not the case. When looking at computer simulations and ancient DNA, this is actually the area where it first arose. So not the UK. So then what can we actually do when we look at invasive species? We obviously can't time travel to look at them, but we have something that's almost as good, and that's herbarium specimens. So herbarium specimens are, well, about 400 years ago, people started to just collect plants and then pressed and dried them and mounted them in such a nice way here and put them in like cabinets and preserved them. So many of those are actually from like the last 100 or 200 years, but we have lots of preserved specimens in herbaria. And especially for common ragweed, you can see here it's quite big, and we only need like a tiny piece, like this big, a tiny piece of the leaf to extract the DNA. And yeah, it has lots of those leaves. So the herbaria were like, yeah, it's fine, just take what you need and do your stuff. It's a nasty plant, we have way too many of those specimens, just use whatever you need. So this is me actually taking a sample from it. And I found it a little bit scary because like my oldest sample is from 1830, which is really damn old. And you don't want to destroy it because other people may want to use it in the future. And because they're so old, you actually need to be really careful when working with them in the lab. So you need to dress up like this. You may know this from COVID, but we already did that before. So you don't want to contaminate your sample. And as you may have seen on the map before, I had quite many samples, like around 600, more than 600. I stopped counting at some point. And like the whole process in the lab for one sample takes about 2 weeks, like from extracting to sequencing. You can do many of the steps in batches, so it's like not 600 times 2 weeks, but still a long time in the lab. So at the end, I more looked like this. So yeah, but then I finally had my DNA and could start analyzing it. So how do you actually become invasive? So the first step, when you get introduced in a new range, you need to survive. It needs to be like the right temperature. Like you don't want it to be like too cold or too warm or too wet or too dry. Like you actually want to find the right conditions. So it depends really like from where to where you get introduced if you can manage to survive. And then well, you need to find a mating partner. You can't be like the lone, well, it's a lion, not a wolf, but yeah, you need to find a mating partner to have like a happy little family and just make sure that the next generation can survive. And that's pretty complicated in the native range because you're pretty alone. And some plants, they just circumvent them. They just clone themselves and they don't need a partner. But ragweed can't do that. It's a lonely plant. It needs a partner. So what it actually can do is when they're closely related species, it can just mate with them and make hybrids. It may not be so good because hybrids are often like, they can't reproduce, or like it depends on which species hybridize. But sometimes, especially for invasive species, this can really help them, especially if those other related species are quite abundant. It can help them, and that's something we actually found with ragweed. So it did hybridize with two other ragweed species, western ragweed here and giant ragweed. And the really interesting thing here is that those two species were actually also introduced to Europe. So they were also not that widespread. So we're not sure how exactly that happened. We want to look into that in the future, but well, it's quite cool they actually did that. And well, it also helps to just have like many individuals introduced at the same time. And that's also something that happened. We know that from historic records that it was relatively many got introduced at certain points. Well, then when you actually manage to have like a few generations, you want to avoid inbreeding. So inbreeding is when you mate with someone that has the same parents, so with your siblings, or also maybe just like share just one parent, or like with cousins. You don't really want to have inbreeding because then maybe something like this happens. Like, look at the royals. You don't want that. So you need to bring in some more genetic variation. And here it also helps when you just introduce enough individuals in the first place, so then you can just mate with someone who has different parents. But what also helps is to have multiple introductions. As I said, ragweed only lives for 1 year, so it only has this one chance to actually find a partner. It dies afterwards. And we know that it was not only introduced once, but it was introduced several times. So then, like this plant here, it was not there with the first introduction. So it brought new genetic variation in there. It's pretty similar to here, but this time the parents just lived on a different continent. And then, like once you're established, you have enough genetic variation, there you can actually also adapt to the new range to be even more successful and spread further. Like ragweed actually does spread further, it comes further north, it adapts to cold, which is bad for you who have hay fever because it gets more common in Denmark, Norway, so I'm sorry for you. So you have enough genetic variation, and well, did ragweed actually have that? So when I look at the native range here, there are actually different genetic populations. So the different colors here, like the blue is different from the orange, different from the green, and different from the pink. So they're different, and 3 of them got introduced to Europe. The green didn't want to. We don't find any traces of this green genetic cluster in Europe, and most of the European ones were from this orange one, but also some blue and some pink ones got introduced. And then they happily mixed in Europe, and now there's like all this genetic variation from those 3 big populations is there. So this is a good thing. There's like then you can actually try to adapt to the invasive range. So we found actually that, not me, it was my supervisors in a previous study, but well, it was found that the European ones, they are bigger than in the North American ones. So there has been some change there, that they actually grow bigger. And also we found evidence that there's some adaptation in connection with defense. So in North America, there are like a lot of pathogens, like bacteria, fungi that can make the plant sick, but also some herbivores that just eat the plant. And some of them are actually not found in Europe. So then the plant doesn't need to defend against them anymore and can just like use those resources for something else, like growing bigger, for example. But there are also other ones in Europe that are not found in the native range. So the plant also needs to adapt to those new enemies. And we found that it actually did that. So it really, within just 100 years, about 100 years, it really managed to change, adapt to Europe, and spread even further. So pretty nasty plant. And yeah, this is it. Yes.
Would it be? Was it possible that birds could have brought it over from North America through eating the seeds and then excreting them afterwards and so on?
Actually, the birds don't really like the seeds, and
so it's just a bad plant all around.
Yeah. It's like a main way of introducing is actually with bird seeds. So the seeds you buy to feed the birds. There's a lot of sunflower seeds in there, and as I showed, like it likes to grow in sunflower fields. So many of those, when you harvest the sunflower seeds, you also have ragweed seeds in there. And when you then import it, the birds don't like the seeds, so they leave them, and then they can actually grow into plants. Yes.
At what point does an invasive plant adapt so thoroughly to its new environment that it's a new native plant?
Okay.
Sorry.
That's a tough question, and then we go to like, what is actually a species? So, yeah.
Later. More beer, okay.
Yeah. Yes.
Did you collect all the samples yourself? Like you traveling all to all this place?
No. No. Also, the nice thing and the bad thing about also using herbarium specimens is they have already been collected, and the herbaria, they just send them to you, and then you can sample at home, basically, well, at the institute, in the lab. But yeah, you don't get to see all those places. My supervisors, actually, they collected many of the modern ones, so they got to see like all those nice places. Yes?
So, from what I understand, you're saying that in the 1800-ish, there was the pink plant and the blue plant just in America. You said the pink species and the blue species and the brown and whatever. And today it has merged into different. So the European was a bigger one, and the North American one was quite a smaller one. Yeah. Yeah. Thanks. Yeah, and I'm just trying to understand, like, so the American one today, has that merged into like a single one all over America, like a single sort of species today?
Okay, this is, all those samples are just one species. Yeah. Did actually everyone hear the question? Yeah. So this is just one species, but there's a lot of variation within the species, also in plant height and so on. But in North America, there was not much change over time from when we looked at. So actually on this map, like the lighter blue is the historic samples and the darker blue is the modern ones. So like the lighter colors are the historic ones. And so it's pretty stable there.
Okay, so that doesn't actually have that much variation in what it It's just the timestamps of when it's dated? Oh.
I don't get the question.
What's the difference between the orange dots and the blue dots?
And the
Are
green
those
dots
two
and
different species?
No, it's the same species, but they're genetically different, so we call them populations.
Okay. That was your question.
Yeah.
Okay.
Oh, sorry. Yes.
Me?
Yeah.
So you showed the example with the hay fever and with example with the sunflower fields for the harming the environment thing. But both of those things are super anthropocentric, including sunflower fields, because sunflower fields are not for the sake of sunflowers, they are for the sake of us liking sunflower oil and seeds. Do they actually also harm the whatever, how to properly call it, like, I don't know, do they kill somebody in the wild or something like this? Like, do they actually harm the actual environment?
Yeah, the thing about like defining an invasive species is like most of them are really human-centered, so we just care about like yield loss and health stuff. But yeah, it's also bad for the biodiversity. So it grows quite tall, and it's actually quite fast that it grows. So then like other slower growing plants, they're in the shadow and they can't really grow then. So it also harms other native species, but there hasn't been that much work on it.
Thank you.
In the back.
How come like when the plant gets introduced to a new continent, how come the pathogens doesn't like travel with it?
Can you repeat?
So the question is like when the plant gets introduced, like how does it come that the pathogens doesn't travel with them? Well, we don't know for sure, but for ragweed, like most of the introduction is like actually the seeds get introduced and not the whole plant. So the seeds come and then they grow in the introduced range, and the pathogens are not on the seeds yet. They would infect the plants when they already grow. Like we only looked at leaf pathogens. So you wouldn't have them on the seeds, and that's why they don't get introduced with them. But some actually could get introduced with them, and also some herbivores. There's actually like a ragweed beetle, it's called, very creative name, because it eats ragweed, and that got later introduced to Europe as well. But at the beginning, you may not have it because there's nothing for them to eat when you still have very few plants in the introduced range. Yes?
Yes, so I actually had two questions. The first question, maybe you are not involved in such research, but I just wonder the population division of the United States populations. So are they divided by natural barriers so that there is no genetic influence, like there's no genetic communication between those populations or whatsoever? So I'm not sure about how those populations are divided so that their genetics are very different. I'm just wondering.
So actually, there's a little bit more going on than what I said. So actually, this orange thing is a mix of the blue and pink one. So probably like the blue, pink, and green were in like different refugia during the last ice age, and then recolonized. And relatively recently, like about 200-300 years ago, this orange cluster formed by just mixing of the pink and blue.
Okay. And the second question is that, from my understanding, you have both historical samples sequenced and also modern sample sequenced. So did you observe any temporal change between the genetics of the historical and the modern samples?
Not so much in the native range. So North America is quite stable, but in Europe there has been like a lot of change. And also like adaptation with like defense and growth and other things.
Okay, so you basically got those conclusions from the comparison between the historical sequences and the modern sequences? Okay.
Yeah, and also comparing native and introduced range.
Great. Thanks.
Yes?
Have we found any practical use for ragweed?
No.
No.
No, it's just a nasty plant.
Fuck this plant.
Yes?
Regarding your answers to one of the previous questions, you said that the orange species were a combination of the blue and green one.
Sorry, I don't hear you.
Regarding the orange species, you said it was a combination between the pink and the blue one. Was there a way to tell if the orange ones like that got introduced to Europe, if it was actually like an orange ancestors, or it was like the pink and the blue which were introduced to Europe and then like mixed there?
So the question is, because the orange is a mix of the blue and the pink, if we can say if the European orange ones are actually, if they formed in Europe, or if actually the orange ones got introduced. So if the pink and blue mixed in Europe. And we have samples from like the very early stages of the introduction, and that's all the orange ones. And if they only mixed in Europe, we would have expected to already then see the blue and the pink. And also, it would probably look a bit different, like, because they would mix in different amounts, and the orange is already like a bit older than it formed in the native range before the introduction to Europe. But yeah, probably we also need to do some simulations to be really sure. Yes.
I'm wondering about the Florida population and why it hasn't come to Europe, because there are microclimates that are like Florida, and could it be, I mean, there must be hypotheses of why it's not found here. Is it sampling bias, or is it that there aren't sunflower or seed crops in Florida that are imported to Europe?
Well, actually, the Florida population was not that well described before we started. But also, like, yeah, in the US, most of the crop production is like in this area and not really in Florida. So you would not really expect that much from Florida coming over to Europe. Oh. Well, my computer says this is over now. Thanks.